individual curiosity, asking technology had Dr. Raji Reddy addressing gathering that was ago 50 years ago decisively moved the for health into the microscope.
Good morning, all. Good morning, all. Hello, ma'am. నేను తీసుకుంటాను నేను టేక్ నేను తీసుకుంటాను
Sorry for the technical interruption. We'll just resolve that in a moment. Thank you.
Hello. Sorry for the technical interruption. This is Mr. Vijay. On behalf of Little Flower Degree College, I take this opportunity to express our sincere thanks and gratitude to a renowned scientist of our country, Dr. S. Chandrasekhar, Director, CSIR IICT, and Dr. C.H. Raji Reddy, Senior Principal Scientist, CSIR IICT, for accepting our invitation to speak on the role of science and technology in mitigating COVID-19 in this webinar. The lectures delivered indeed by our speakers were very informative, inspiring, and motivative. So I request Dr. Gomati to read the profile of the college. And I request Mr. Raman to play a short video about our college.
Little Flower Degree College, Uppal, Hyderabad, a Montfort institute run by the Brothers of St. Gabriel Educational Society, has been illuminating the world around with its motto, Lead to Illuminate. In its 12 years journey of excellence, LFDC has been fostering the young generation to be humane and preparing them to compete, realizing their dreams and purpose in life on the competitive global platform.
Good educational environment, discipline,
practical skills helped me in getting my first job.
Excellent placement opportunities.
LFDC is an awesome college.
Dear parents and students, Little Flower Degree College, Uppal, Hyderabad has begun its admissions for the academic year 2020 and 21. The online application for the 3-year degree in BA, BSc, BCom and BBA is now available on our website.
Little Flower College.
LFDC.
Little Flower Degree College.
Thank you, Mr. Raman. Now I request Dr. Gomati to
It's my honor and privilege to introduce our today's speaker, Dr. S. Chandrasekhar, Director, CSIR IICT Hyderabad. Dr. S. Chandrasekhar completed all his primary and higher education in Hyderabad and joined CSIR Indian Institute of Chemical Technology for PhD program. After completing his PhD with the then director, Dr. A.V. Ramarao, he moved to USA for a postdoctoral position with Professor J.R. Falk. He joined CSIR Indian Institute of Chemical Technology as Scientist C in the year 1994 and grew up to the level of the director in 2015. He is a fellow of all the three Indian Science Academies, that is, National Academy of Sciences, Indian Academy of Sciences, and Indian National Science Academy. He is also an Alexander von Humboldt Fellow. He has made significant contributions in diverse areas of organic chemistry, with a special emphasis on chiral chemistry, total synthesis of biologically active natural products, and pharmaceutical products. He introduced polyethylene glycol as a novel, environmentally benign solvent medium. He has developed technologies for the synthesis of latest anti-tuberculosis drug, bedaquiline, anti-tumor and abortive drug, misoprostol, antiplatelet molecule, Beraprost, antidepressive compound, sertralin, and drug for treatment of schizophrenia, asenapine. He has more than 275 publications with 6,500 citations. 65 students have been already awarded PhD degree under his able guidance, and 20 postdoctoral associates have worked in his group. He has received several accolades, including Eminent Scientist Award for the contributions in the field of chemistry from Telangana State Government in the year 2017, CNR Rao National Prize for Chemical Research in 2012, CSIR Technology Award in 2014, and Infosys Prize in Chemical Sciences in 2014 for his contributions in synthetic organic chemistry, with special focus on the synthesis of complex molecules from natural sources and innovative practical approaches to pharmaceuticals of current interest to industry. Little Flower Degree College cordially welcomes you, sir.
Now I request Dr. Chandrasekhar to take over the session.
Good morning. Can you hear in Little Flower College?
Yes, sir, we are able to hear you, sir.
Perfect. Okay. So good morning all, and I see about 290 participants on call. I invite all of you to the session. I also would compliment Jayanti for taking this initiative to connect scientists to some of the students to tell them what is happening around the world currently today with science and technology on COVID. All of us are suffering for sure for the last few months because of COVID-19. And lockdown is not an easy job. And now we realized when someone is given imprisonment, rigorous imprisonment for 6 months in a jail, now we know the kind of mental trauma a person who commits a crime will undergo. So this is no less. We don't know the crime we have done, all of us, the entire planet, and the whole world has got this punishment. But when you really look at the scenario, many theories have been coming up for why this SARS-CoV-19 has erupted. There are many scientific discussions, logics, computational models, and many things have come up. But I don't think any of those models or predictions are 100% correct or they're 100% wrong. People have been telling that because human race has become very selfish and we have been taking the space of animals and plants, and in the process, the animal kingdom got extincting, and in the process, some of the viruses have erupted in bats and they have come back to the human beings. While all of us know that bats are the primary hosts for many viruses, and these viruses don't harm the bat because bats don't have receptors that they will go and bind and replicate. But we should know that science and technology has handled this pandemic much better than previous pandemics which have occurred on this planet before. We all know that 1918, the huge pandemic which has taken several millions of lives, which was also called a flu pandemic. Today is also flu pandemic. So it's exactly 102 years, and we got the pandemic which is almost as large as that. But in between, we have many eruptions of diseases. They could be bacterial infections or viral infections. Viral are more dangerous because we don't know how the viral spreads happen. Many times we know that bacterial infections happen from person to person. But if you look at some of these viral disorders this human race has encountered in the last 100 years since we started recording science and technology are many. Thus, it is almost very certain that pandemics do happen to human beings, and human has to learn how to live with them. We have seen problems with polio. We have seen problems with smallpox. We have seen problems with chickenpox. We have seen problems with HIV. And we have seen Ebola eruption in 2009. We have also seen bird flu, and many infections triggered by virus are not new to the human race. So we need to go back and see what is that new this corona COVID-19 has taught us more lessons than the previous pandemics. One, because of social media, we get information much faster than it used to be in early 2000. Was it a good news or a bad news? I think it's a bad news, because now we are seeing enough jokes on WhatsApp and Facebook that WhatsApp has become the most prestigious university, sharing everything which is not proven scientifically. Someone who can write good English or someone who can cook some proper tic-tac photographs or videos can dump on social media to create more panic than the actual disease is. It does not mean COVID is not dangerous. It is certainly spreading very fast, but it is not spreading as fast as the wrong news which is spreading on the social media. Because I'm talking to some of my young friends in college, I thought we should know how to use this social media to our advantage and not to spread things which are not validated. What is validation? Unless you do a scientific evaluation and do some conclusions which are having a logic, only those news should be shared with friends and colleagues. But unfortunately today, someone would write on social media that banana carries COVID-19 and we will stop eating banana. Someone will post that the virus will live on the surface for several days, and then we trust it without any scientific evidence. So I thought I will take this opportunity to talk to some of these young minds and tell them that don't carry away by these half informations, which are mostly lies. So today we are living in a world where even truth has to be justified. And if the person who knows the truth, for example, a scientist or a medical doctor, does not have proper communication skills, but he knows the truth, he cannot convey the truth properly. But someone who believes a lie and then he wants to spread the lie, he has very good communication skills. I think he can communicate the lie much better than the truth has been spread by someone who does not have communication skills. Thus, many news we hear under this scenario, I tell all my young students on this forum that please do the fact check before even we believe that what the news we are hearing is correct or wrong. You look at many television channels and very populous or some titles which attract the media or the public are flagged. And a very small problem sometimes is shown as a large problem. So as a scientist, I thought I would tell my young students that they would go back and tell their parents and also communicate to their other friends who are not part of this session today that we should not spread false information just because you have a mobile phone with unlimited internet connection. So this is one we all should take seriously. Again, when COVID-19 erupted in December in 2019, last week of December, we all should be proud of scientific community and scientific achievements the world has achieved in the last 40, 50 years. That within less than 8 weeks of time, the scientific community is able to solve the genetic sequence of this COVID-19, to design what kind of vaccines or what kind of drugs one could quickly discover and stop this pandemic. Within no time, the scientific community has realized that hand sanitization and social distancing will minimize this problem. If you go back and see the flu of 1918, at that point of time, because science was not well developed, we were not aware of social distancing, nor we were aware of how that flu was spreading. That was the biggest cause where several millions of people lost their lives. If you go back and look at the 1918 flu pandemic, I will not be exaggerating, more than 20 million people have lost their lives. However, when we got the bird flu a few years ago, or when we got Ebola, or when we got swine flu, all these flus are cousins of each other. We were able to make drugs available at the earliest time. We also understood how the disease is spreading, and we take all the precautions that minimal lives were lost. While we also handled the viral problems very well, all young students must also know that whenever cyclones and tsunamis were hitting the planet, because our geophysical predictions were not at the best those days, we used to lose many, many lives. We also know those who are from Andhra, near Vijayawada, when there was a cyclone, villages after villages were wiped out because we were not knowing when a cyclone would hit. Similarly, we have seen many, many cyclones which have taken lives before 2000. But today, the predictions we make based on mathematical models, the satellite pictures, we know that at what speed a cyclone is moving in the sea, by what time it will hit the land. And government takes all precautions to make sure that no life is lost. We all make sure that every human being on the coastal part is lifted and taken away from the shore so that we don't lose any lives. Today, when we look at many, many cyclones we see today and tsunamis, we see hardly we hear any human life loss except the property loss. Thus, science has helped how to move people well in advance. Similarly, today, if you look at the pace at which science is growing, we are confident by next 1 or 2 decades, with brilliant minds like those who are on the call today and trying to get inspiration from science and technology, I do believe all of you will get an opportunity to join the scientific community and do science which helps humanity. We all know that India has beaten polio and we don't see any polio patients in our country currently. Similarly, we have eradicated totally the smallpox. So scientists have a power, they have knowledge, and COVID also will lose the battle sooner than later. Thus, we need to respect the scientific community and make sure that scientists also practice ethics to make sure that only truth, logic, and rational prevail, and we don't go with anything else. If we look at how the COVID was spreading, we also looked at initially this disease was spreading from Wuhan, a big city in China with over 1 crore population, and many flights were going out of Wuhan to Europe on business purpose, and that is how this virus has spread to Europe. And when people were coming from Middle East, Europe and America to India, that is how we also started getting this. And the first patient we recorded in our country was in Kerala. So far, India has done well with proper social distancing, lockdowns. We were able to see that number of cases and number of deaths are minimal. There are many theories which are floating that because Indians have innate immunity, which is what we acquire as we grow because of less hygiene around our surroundings, we might have got some acquired immunity, and that is helping us to fight corona. Also, there are also predictions that India has a BCG vaccine, which is given to all children when we are young. And maybe that BCG vaccine has produced some antibodies, and these antibodies are giving us some kind of a protection. We only wish that this corona will not trouble us so much as it has troubled Italy, US, and few other countries. We are also fortunate in a way that it's a late entry into our country. So we got enough opportunity to prepare our country to make sure that we have enough hospitals. We will make enough PPEs, personal protection equipment for doctors. Hopefully in the next few months, we may also get enough drugs. Who knows, the clinical trials happening with the help of Cambridge University, Oxford University, and also with one of our Indian companies in Pune, Serum India Limited. If a vaccine could come by December, January, I'm sure we would be able to handle this pandemic much better than other countries. But as youth, I earnestly advise all of you, as we come out of the lockdown, please don't believe that lockdown is lifted does not mean corona has left the planet. It is the government which has taken lockdown to make sure that enough social distance is maintained. India gets ready to handle the patients when they increase if the pandemic goes much larger. So government has used this 3 months of time to make sure that hospitals are made ready, all the drugs are made available, like azithromycin or hydroxychloroquine, or whatever drugs doctor would like to prescribe, to make sure that India is ready. And then in the meantime, we also learn to live with social distancing. We all know that India is one of the densely populated nations. And in our country, to keep social distancing, we all need a very serious commitment. Please see that some of the people who are not educated like us, we are all fortunate that we got opportunity to go to colleges and schools. There are several underprivileged people who are not able to go. I think as students of science and scientists, we have a moral responsibility to educate these people the importance of social distancing and the importance of hygiene. We all know that our palms carry the virus when you touch some surfaces. So how frequently we wash our hands? How do we help some of our relatives who live in small villages who have no access to sanitizers? how do we prepare sanitizers? I'm sure with the availability of internet, you can go back and see how isopropyl alcohol or ethyl alcohol can be used as a sanitizing solution. So can, as science students, we help some of these people who are not educated or who have not gone to science education to help them to make their sanitizers so that they protect themselves from this pandemic infection. Can we also help everyone by encouraging all our neighbors to mandatorily wear a mask? Scientifically, it has been proven that if someone who is a symptomatic or asymptomatic positive COVID patient wears a mask and someone who is talking to him also wear a mask, the chances of when the person coughs, the droplet traveling and going through the nasal sacs or oral route to spread the disease comes down drastically, and there's only 2 to 3% chance that he will spread the disease. If both don't wear a mask, the chances of spreading is more than 95%. So, as science students and scientists, it's our responsibility to scientifically educate all our neighbors and friends who are not aware of hygiene and social distancing, is to see that they follow this very, very carefully. As a scientific community, entire India, especially the Council for Scientific and Industrial Research, where IICT is one of the laboratories, CCMB, which we are neighbors to Little Flower College, and many other labs in the country have risen to the occasion. When all of you have gone into lockdown, we did not take the lockdown. Rather, we took the risk of working in the lab with all our students, of course, with proper precautions like social distancing and wearing masks, and made sure that in the lockdown period, we develop enough technologies. We all have been seeing that India does not have enough ventilators. For example, oxygen ventilators when patients have really gone to serious and admitted in the hospitals. Personal protection equipment for hospitals. We have a lab in Bangalore called National Aerospace Lab. So this lab, taking advantage of how we use the PPEs for satellite, spaceship and all that, the knowledge the aerospace lab has, we went on to make now PPEs. Today, our industry partners are in a position almost to make 50,000 PPEs per day, and we are scaling up to 2 lakh PPEs per day. Similarly, CCMB, which is our neighboring biology lab, started screening the patient samples. Those who are students of biology know that it is not easy to handle patient samples, especially if they are corona positive, because you may get infection with that. So we need to have a BSL3 facility, which means biology safety level, biosafety level 3 facility, which works under negative pressure to see that when you are in that room, when you breathe also accidentally, you should not be able to take the breath inside. So there's always something which comes out of you into the system, but nothing comes from system into your body. And wearing complete protection of the nose and everything, one goes into this chamber and work on patient samples to test whether the sample is positive or negative. So CCMB scientists now today do almost 4,000 samples of patient samples. While doing that, our lab also went on to sequence the genes of this virus to see whether this problem is very different, whether the Italian sequence is different from Indian or American or Chinese or Middle East. So this opportunity helped our lab to understand whether the COVID what we are getting is different from other countries. Similarly, IICT, to which I represent, and Raji, who also will talk to you, how we were able to discover new drugs in less than 2 months and see that they reach the patients. while it is impossible to discover new drugs, what we have done is, if already existing drugs in the global market, for example, we all know that when we get headache, we take paracetamol or aspirin. But we also know that if you take paracetamol, the body temperature comes down. So when a COVID patient gets high temperature, whether paracetamol can be used, whether we have enough stock of paracetamol in the country or not. Similarly, we all know that whenever we fight with viral infections, we all get secondary bacterial infections. We all know that virus has no cell wall, whereas bacteria has a cell wall. So there are two different classes of living beings. Virus per se does not live on its own. Virus always wants a host to survive. It's just an RNA sequence, a small chemical comprising of nucleic acids. And this RNA goes and binds onto human cell or animal cell. And it takes the entire control of the machinery of our cell. Because our cell has a role to play for all of us to live and perform our duties. So cell does its job, but when viral RNA sequence come and control our human cell, it will start giving wrong instructions to the cell. And now those problems will get into high temperature and breathlessness problems in the lung and few other problems. So there are several targets in our human cell where this COVID virus will come and take shelter. So what scientists will do now, what we are trying to do, what places on the human cell this COVID-19 gets peace, that this fellow come and start enjoying our cell and taking control. So with the help of some chemicals, can we eat some chemicals, which we call them drugs, that these chemicals will go and sit in the cell and will not allow the virus to come and settle in the cell. So we all saw in the papers that this corona is called corona because it has a crown protein. So this protein structure is now solved by scientists within 2 weeks of time, which was not possible 30 years ago with no science in the world. So we were able to solve the structure of coronavirus. The protein which is encompassing this corona, the crown structure, the spike protein what is on the coronavirus, and where it goes and binds into our human cell. So we went on to make sure that these human cells will not become vulnerable and surrender to the coronavirus. So this is how we made sure that the drugs will come. Raji will talk after some time on how IICT has made two major drugs. Favipiravir, which is an antiviral drug for common flu, which is used extensively in Japan. Then remdesivir, which was discovered by a company called Gilead in US for Ebola virus, but it's been repurposed. Repurpose is what? It was discovered for a particular purpose, but knowing that that disease and this disease have some similar patterns, whether we can repurpose the drug discovered for some other disease to the current problem. So we were able to repurpose some of the drugs and see how best we can handle. We are very happy to tell all my young friends that in the next 8 weeks, multiple companies like Cipla, Glenmark, and few other companies would launch, once the clinical trial data come up, some drugs which will help us at the moderate cases and early cases of the disease. When someone gets early symptoms and they become corona positive, even before they are admitted in the hospital, if this drug is given, the chances of cure becomes much better than without any drug. We all should know that there is no drug available currently for corona treatment except remdesivir, which was approved by the US on an emergency basis. Also, vaccine clinical trials are happening globally, and we all know that a drug discovery cannot happen just like that. If someone wants to discover one new drug, you please know that someone who knows chemistry should know that we should make about 100,000 new chemicals, test them in cells, test them in animals, small animals like mice and rat. We should test them in monkeys and chimpanzees and dogs. And then we should do phase 1 trial, phase 2 trial, phase 3 trial. and it takes about 8 to 12 years to get a drug for any new disease. So in that context, if we know that corona disease came onto this planet only in 2019 December, remember we need 8 to 10 years before a drug come in. But virus also has problems that it keeps undergoing mutations quite often. Thus, we need to keep a track of what kind of mutations it is happening so that we design our vaccine studies in that fashion. So don't carry away that a vaccine will come and then we don't require to do social distancing and all that. I think we need to be extra cautious, especially a country like India where it's a very crowded country. People in Hyderabad, we have a lot of crowds. So please make sure that whenever you see friends or colleagues walking around closely, we should go and tell them, please keep social distance politely. Many times people wear a mask in the neck, like I'm wearing currently. But because I'm alone in the room, I can wear like this. But the moment you start going out, please wear the mask in such a way that you don't expose your nose and mouth to someone who is talking to you without a mask. Also, if someone is trying to talk to you without a mask, please make sure that enforce him to wear a mask before he starts talking. So if we can make sure that both people wear a mask, the one who's talking and the one who's listening, I think the chances of corona spread would be very, very minimal, and we can have a very safe go till the vaccines and drugs will come into market. We also believe that if the coronavirus undergoes mutations which become less virulent, we'll be lucky. But if we are unlucky and if they become more virulent, then we have to be extra cautious. So it's a message to all my young students, about 340 participants online, that please take the responsibility of educating people who are not in science background to make sure that while drugs will come, it takes some time. We also will tell that social distance is the only mantra to avoid this problem. When common cold and flu season starts, once this monsoon sets in and rains will begin and winter will start, it will become very difficult for all of us to differentiate between a seasonal flu versus the COVID flu. We believe that before that happens, we'll be able to curtail and India will get a flat line like Europe. Today, we know that Italy, France, and all have got a maximum hit. Now, slowly there's a decline. And hopefully, India will also come up with some solutions in the meantime, that without many human losses, we will come out of this problem. We also know that people with diabetes, cancer, and hypertension kind of people are more vulnerable. So many of us who get COVID-19, if we are generally healthy, within 14 days, the viral load will come down and will become normal without any medication. However, if some other diseases are there, there's a chance that they will become serious. So it's also our responsibility that if our grandparents and grandmothers and any of our uncles and aunts who are older and they have some diseases, it's our responsibility to see that they are kept social distance in a private room, and those who are going to places for shopping, for grocery and stuff like that, they don't go close to those people, because it is the interest of our own family. We should make sure that our uncles and aunts and grandparents will not be affected because of the problem. As I told you, we are also doing a clinical trial on another vaccine in India, which is called MW. And we also believe that this vaccine trial also will be over in the next few months. And we all believe that by December or January, in the next 6 to 7 months, hopefully some vaccine will give protection to us, and also some medication will come, and social distance. So these three characters, social distancing, medicines, and vaccine put together, we can defeat corona, and then we can also go back to normal life very soon. But if you are ignorant, like today we have seen many of the workers who are migrating from one state to other state, unfortunately, this again is spreading, and number of cases have gone to 10,000 today. Had we taken a very strong decision of not allowing our migrants, maybe we would have controlled faster. But unfortunately, these migrants were stuck away from their families, and they had a lot of family pressures, and that's forced them to go back to their families. So it is time that we do social distance. We also educate our people. And then if the spread is becoming slower, in the meantime, I can assure the scientific community will give enough drugs and vaccines to defeat this problem. Also, when people get COVID positive, we should tell our friends that they should not get panic. It's a disease where almost 85 to 90% of the people who get COVID positive, within 7 to 14 days, even without any treatment, they will become normal. Only 3 to 5% people need hospitalization. And after going to hospital, they will be treated with some antibiotics, some antiviral drugs, like as I told you, favipiravir, remdesivir, and some other virs. Actually, in education for all students, any drug where the end letter is VIR, that means they are antiviral. Any drug name which ends with AB, then those drugs are called antibodies. So these are the things I think a scientist and scientific student should know, that any drug whose name ends with VIR, they are antiviral. Any drug which ends with AB, they are imitinab. For example, IMITINAB. That means it is an antibody which is generated to cure some diseases. Many antibodies are there for cancer treatment. Today, we are also trying to see whether those antibodies can be injected to COVID patients to see that COVID can be treated well. Also, immune boosters. If we can, our immune system is good, we can defeat COVID very easily. So malnutrition is another problem. So I think all young students, eat well, eat nutritious food, don't eat garbage, keep hygiene, and I can assure you that we can handle this problem very, very easily. And in the meantime, you give us some time to scientific community. As I told, drug discovery takes 8 to 12 years. But currently, the way scientists are working, within 2 years or 1 year only, we can make more drugs and we can handle this problem very, very well. I wish all of you safe stay at home and use this stay at home reading literature on science, not fiction, not some things which excite you temporarily, but please read scientific literature, something which is useful to your subject, that all of you who are listening will come into scientific community in the next 2-3 years, join the scientific force. Science is looking for bright minds, and all the bright minds, if you join science, it is not difficult to defeat these viruses. Please remember, COVID-19 is not the last virus this planet will encounter. Who knows, every decade or every few years or every 100 years or every 2 years, we don't know how frequently these endemics and pandemics will keep attacking. We all know that when bird flu came, we all thought that bird flu will come from chicken, and we all started killing all the chick. Then someone says swine flu, we started killing all the pigs. If tomorrow someone says a disease is coming from human to human, do we kill human beings? Can we kill fellow human beings? Today we know that COVID comes from human to human. I think killing is not the solution. Getting drugs is more important. Developing more vaccines is important. And to develop vaccines and drugs, we need bright students into scientific community. And I'm sure that this last 40 minutes of webinar will motivate some of the young minds to take up science as profession. I'll be happy to take some questions. I think they are being typed on the sidebar. I will keep trying to answer, but I can ask moderator to ask if moderator has anything to comment. Thank you, Jai Hind.
Yes.
Thank you, sir, for
Thank
such
you,
an
sir.
interesting and informative session. We got a detailed information of the pandemic mitigation, and many of us students will take up science and become scientists like you. Knowledge is having the right answer, but intelligence is asking the right question. Now I invite my colleague, Dr. Gomati, Head, Department of Zoology, to conduct the questionnaire session. Dr. Gomati.
Thank you, Mrs. Rashmi. That was indeed a very vivid presentation. Our participants would have got enthralled in the session. As Dr. APJ Abdul Kalam had said, learning gives creativity, creativity leads to thinking, thinking provides knowledge, and knowledge makes you great. There is a huge response from the participants, sir. I would like to present their queries. Sir, what will be the impact of the virus in the post-lockdown?
we have already seen the post-lockdown, the number of cases are increasing. And we have seen that initially we have reached a stage where doubling of number of cases was taking 14 days. Again, we came back to 12, 13 days. So post-lockdown is created because people were losing jobs. We wanted to make sure that people who are stuck at their hometowns and different places have to go back to their workplaces or hometowns, vice versa. So that is why we have lifted the lockdown. Lockdown does not mean virus has left the humanity. Still the virus is around us. So social distancing, hygiene have to be kept on top priority when we go back to work. Today, IICT has opened up. We were working at 30% staff in the last 2 months. Currently, we are operating 100%. Now, we do differential timings. So when you open the college, I'm sure you also need to do something that each classroom will have only 15 students. Every bench, only one student sits. Everyone wears a mask, mandatory. Teacher wears a mask. And hygienic, your sanitizers are kept everywhere. I think we cannot permanently live on webinars. And we cannot have exams at home. So certainly in the next 1-2 months, we need to open up, especially colleges and schools. But one blessing in disguise, young population, students in the age group of 17 to 20 are less likely to be affected badly. They will get disease, but they have more resistance to fight. Does not mean they should go on the roads, because they may become asymptomatic carriers. For example, if your BSc first year boy or BSc second year boy come to college and he carries it somewhere in auto rickshaw or a bus stop or a shop where he buy a peanut or a mango or something or chocolate, and then he may take home, and if he may not show any symptoms and he thinks he's normal, but he's carrying, and his grandparents who live with him in their house, they may become victims and they cannot survive because they are old and they have other elements. So youth should be very, very cautious in the post-lockdown era.
Sir, next question is, as the virus is evolving very dynamically and able to manipulate its surface structure, so its stable structure, how will we be able to identify, and is there a comeback during the winter again?
There is nothing like comeback because it's still with us. It has not gone away to come back. It is still with us. Okay. And please remember, Ebola virus is also there in the system. Okay.
Okay.
But whether our body will develop a native immunity over a period of time, and then these viruses will not trouble us till a new virus comes.
Okay.
So basically, those viruses will not disappear from the planet. They may be in some other host. They may go in human we control, but they will continue to live in bats, for example. And then they may come back. Similarly, swine flu. Do you think swine flu is gone? But we are not scared of swine flu. When bird flu came, we were all scared. Today, we are not scared of bird flu. So once the medicines come, vaccine come, and about 70-80% population will also get immunity, then naturally the carriers will become lower, and then the fear will go from our minds.
Okay. Sir, remdesivir, the drug designed for hepatitis C and Ebola, now a potent drug for COVID-19, what will be its target site?
Basically, they will not allow the RNA to replicate. Okay, because RNA would like to go into our body and it starts replicating. So they will stop the replication. So what we tend to do is, we create a drug combination. Anytime a viral disorder comes, we always develop cocktail drugs. So you make one drug which will stop replicating the virus. We also give another drug which will go and sit in the human cell, and which will not allow the virus to settle in your cell. And we also give a third one which will immune boost. So many a times in viral disorders, you give combination of treatments. That is how we have controlled HIV. I'm sure many young people on the call may not know, but some of the teachers in 87, 88, when HIV was a big disorder, which just erupted in 87, 88, if I'm not wrong. I was a student at the time. So people were scared to touch each other because HIV will spread from human to human, like what we were talking today. People were scared to barber shop because when you go to barber, when he shaves or uses some blade, the blood transfusion will happen, HIV will come. And HIV is a disease where it works on the human immune system. It weakens immune system and people will die. So you can't get any drug for HIV. Today also we have more than a dozen drugs, but they don't cure HIV. They only manage HIV. Like diabetes, we only manage the disease. We don't cure diabetes. In the case of this influenza virus, which is a RNA virus, we believe that once we develop some vaccine, 70-80% of the people will get protection. Like if you take every year a flu vaccine, more or less we all don't get a common cold and flu symptoms very often. Yearly, some people get 10-12 times. But if you take a vaccine, you may get only one time. So we believe that vaccine and drugs will help us and then we develop more and more RNA-based drugs.
Yes. So the next question is, how effective is plasma therapy in the treatment of COVID-19?
It is working well because what they do is all of us know that whenever a new virus attacks our body will react and produce antibodies. And those antibodies is the fighting mechanism by which we can eliminate the virus. So someone whose immune system is not very good, they may not produce sufficient antibodies. So what you do is someone who already got a COVID-19 disease, you take plasma from them so that it will contain a lot of antibodies. Those antibodies you inject to the patient whose body is fighting, but you enhance those antibodies in the body so that the patient gets cured faster.
Yes, sir. Sir, one last question. How do you think that whether the rupee note, that is rupees, whether it will carry the virus?
Actually, there is no very proven scientific evidence that surfaces and the paper and all carry virus for too long. It's always good to avoid touching any new surface. Okay, but
Yes.
anyway, we are going on to Paytms and we are going digital cash transactions anyway post demonetization. So in case we are scared of it, what I suggest is if any currency you touch, immediately you wash your hand. I think that's something which you should do as a precaution.
Yes, sir. Great teachers emanate out of knowledge, passion, and compassion. Our distinguished teacher also patiently answered to the queries. Thank you, sir, once again for answering so patiently and giving a lot of information to our participants. Thank you, sir. Over to Mrs. Rashmi again.
So I'll sign off because a few other things to do. Good luck to all my young students. God bless you and make sure that you keep proper social distance. I've seen youngsters believe that nothing happened to them. Please believe virus will not spare any of us. So the only solution currently is social distance, wearing a mask. So these two you please follow carefully with personal hygiene. And when you go home, make sure that you leave your chappal outside, wash your hands immediately so that anything you touch, you don't carry virus in your home. You may not get disease, but your grandparents and uncles and aunts are old at home. You being going out more frequently, you will carry back home. So this precaution you must take till we get a cure. Thank you all.
Thank you,
Thank
sir.
you, sir, for efficient handling and clarification. Our students will really take all your words into consideration. Thank you, Dr. Gomati. I would like to invite Mrs. G. Srilekha, Head Department of Physics, to introduce our next speaker of the day, Dr. Raji Reddy, Senior Principal Scientist, ISCT, Hyderabad. Dr. G. Srilekha.
It's an honor and pleasure to introduce Dr. Raji Reddy, a principal scientist working in Natural Products Chemistry Division, CSIR, IICT in Hyderabad. Dr. Raji Reddy graduated in Organic Chemistry from Osmania University in the year 1997. He is awarded doctoral degree in the year 2002 in the field of Organic Chemistry from CSIR, IICT, Hyderabad. From 2002 to 2005, he completed his postdoctoral research at Department of Chemistry, University of South Florida, USA, and the Department of Medicinal Chemistry in University of Mississippi. After accomplishing his postdoctoral research, Dr. Rajareddy served as principal scientist and senior scientist in many prestigious organizations and currently serving IICT as a principal scientist. It is indeed a matter of honor to know that more than 10 students were awarded doctoral degree and many are actively participating and pursuing research work under his guidance and supervision. He has authored more than 100 research articles and in the most peer-reviewed national and international scientific journals and currently holds 3 patents on his name. His list of awards includes, but not limited to, Young Scientist by AP Academy, AVRA, and A.K. Singh Memorial in the year 2007, 2011, and 2014, respectively. Best Performance Award in the year 2010 by IICT. Best Lecture Award in the year 2011, and Associate Fellow in 2013 by AP Academy of Sciences, and many more. We are extremely honored and privileged to have you as our guest speaker for today, sir. Little Flower Degree College welcomes you.
The science of today is the technology of tomorrow. And technology is best when it brings people together to make them understand that however difficult life may seem, there is always something you can do and succeed at. It matters that you don't give up. I welcome our next speaker of the day, Dr. Raji Reddy, Senior Principal Scientist, ICT Hyderabad. Welcome, sir.
Very good afternoon to distinguished lecturers at LFDC and also all the participants. Very good afternoon and welcome to this webinar session. First of all, let me thank you, Ms. Srileka, for kind introduction, and also my thanks goes to LFDC management for inviting me to this webinar series. Hope I'm audible.
Yes, sir, you are audible.
Oh. So, our beloved director, Dr. Chandrasekhar, gave a brief overview of coronavirus and the treatment of COVID-19 and all other important things about the virus. So, what I thought of doing, because our specialization is organic chemistry and we are working in the synthesis of these drugs and pharmaceutical, I thought of educating these graduation, post-graduation students how this organic chemistry plays a key role in drugs and pharmaceuticals and various other things. In between, I may show some of the slides if possible, but all of you know that chemistry especially plays a key role in all our day-to-day life, and any activity, there is a chemistry and reaction is involved in all activities. So in a similar way, for discovering drug or a pharmaceutical, also this plays a key role. As in the earlier lecture, you have already heard that drug discovery takes 8 to 12 years, and each year a particular activity goes on, starting from the identification and synthesis of various chemical compounds, thousands of compounds. So how to make these compounds, how this has evaluated over the years of period, and what is the present situation, and how we applied this knowledge throughout the 100 years today for COVID-19 treatments. So in olden era, all of you know that how the drug discovery, olden days, how people used to take medicine. So what is that major important thing people used to use is a natural product, whatever available in the nature, and based on the traditional knowledge, they used to take that traditional plants or other things in a raw, crude way. So that's where the drug discovery began. Now I try to share some of these slides so that it will be. Are you able to see?
Yes, sir.
Yeah. Yes. So this is how I will just give a brief so that all students will understand how the drug discovery in general. I believe most of the students are in science. Science students are participating in the webinar. So particularly the olden days, the drug discovery used to be, you can see from this picture, olden days when long back, this is how we used to treat various kind of problems, especially these natural products, mainly as a medicines described throughout the history. And this is where we will take as an initial knowledge. This is how it started. And then natural products means it's not only plants, there are various things available. We can see that the microbial world, lot of fungi, algae also will produce lot of compounds. And of course, plant sources, the best example is the paclitaxel, which is from Taxus brevifolia, currently used for breast cancer drug. It originated from this particular plant. And we also have marine sources. A lot of compounds will be produced from the algae fungi under the sea. And there are few compounds were isolated and used as drugs even from animal sources. So this is a broad way of classifying the natural products. And then this is the drug discovery history, natural advantages, because it has a great diversity. The nature selectively produces compounds. So that's why it's an excellent source of novel compounds, so that we can understand the mechanisms and other things that will majorly guiding our drug design and other process. So that the present medication mostly depends on the natural products, either directly or indirectly. So you can see that according to even WHO, about 80% of the population relies on medicine still. So far, 121 drugs described in USA today come from natural sources. So that's, as I mentioned, either it directly or indirectly from the plant sources, and 47% of the anti-cancer drugs in the market come from natural products. How you can see some of the examples. This is the one of the oldest one. You can see that morphine, and it was long back, and this was used for treating malaria and asthma, diarrhea, other things. And many of you know how the drug discovery, natural product, how it is helping. Aspirin is the best example. One can see that. So while no one person invented a drug, it is a team effort. And how just a small couple of slides, you can see that how aspirin was discovered. It was actually a natural product source. This was a tree long back. People were using in 1700s. A scientist, Reverend Edmund, was using this white willow tree for the treatment of fever and pain whenever the temperature is there. But what they found is it was very bitter, and then the chemical known as salicin in this. So this is what salicin. But when they are taking this as a for the treatment, either reducing fever or pain, and then they found that it is causing some ulcer kind of thing. So for that purpose, for many years, this was used as a medicine. But later, what happened? One of the scientists from the Friedrich Bayer company, Felix Hoffmann, who worked in this company. So when these chemicals using the stomach upset and other problems, and then he decided that what is that we should do for this compound. And from that, it was found that one of the hydroxy group, you can see that was protected as acetylated, then it became an acetyl salicylic acid. Now, once this hydroxy group is causing for acidity, when it was protected, then it was discovered as an aspirin. So this is just simple one example how natural products are the basic origin for
Can you stop?
So sorry to interrupt. So can you hear us?
Found that a lot of side effects by taking this drug. So in many places now it was also banned, but in some places it is being used. But this is only just for example. I have shown here how natural products are very important in discovering a drug. Now, if you see the history, see the first example I have shown the acetylsalicylic acid from salicylate, and then you can see the morphine, then you can see the complex structure digitoxin, then quinine, and then pilocarpine. So these are some of the natural products became the drugs into the market, and quinine was approved by FDA in 2004, which was isolated from also one of the tree. Then you can see all many of our antibiotics, penicillin, narcoticin, imipenem, aztreonam, all these are natural products, and you can see the structures, how complex are they. Then the next generation, even for anti-cancer drugs, doxorubicin and paclitaxel, these are also all isolated initially from natural products. And you can see how the complexity is increasing. These are the presently one of the very effective drug for cancer, breast cancer, particularly this ergotamine mesylate. It was also originated from this left side part. You can see here olicandrin B, a marine source. It was isolated, and part of it became a drug as a ergotamine for the treatment of breast cancer. And this compound we have also made in IICT in a couple of years back. And you can see the list of natural products, how they are, helped in drug discovery, either as such natural product or in the form of their derivatives came into the market for various disease treatments. And this is just a graphical picture where you can see that all the new chemical entities like NCEs are from natural product derived compounds. Now, let us little bit switch into organic synthesis. Now we know that natural products are sources for drug discovery. But you just imagine how much quantity of aspirin or how much quantity of any medicine we require for whole world population. Requires some of the cases thousands of kilograms, or some of the cases in ton scale. Suppose if you take paracetamol, how many tons it required all over the world. So it's a huge amount. And if that is the kind of amount, if it is a natural source, you cannot extract everything from the natural source. The one way that we cannot actually get that much amount of natural sources to extract, or also we cannot destroy the nature. Now nature showed us a way that this is what compound it is useful for the treatment of a particular disease. Now what we have to do, that particular structure after identification, we have to make that in the laboratory. That art is called organic synthesis, and whatever you are making, you are using the medicines today, all made in the laboratory, either initiated from natural product. But unless you make in the laboratory, you cannot have whatever the quantity we require. So you can just see the organic synthesis plays a key role for various things. What is chemical synthesis? Intentional construction of a molecule. So we know the structure. We have seen several structures in a couple of slides back slides. And now knowing that structure, how we can make in the laboratory, we have to plan. That is called a synthesis. So why we need synthesis? As I told, because from nature we get limited quantities. That is why to make more quantities, the synthesis plays a key role. Other than making quantities, it also helpful in other things. It inspires chemists to develop new reactions based on the structure, and also it helps. So if you go to history of synthesis, you can see this is the best example. The first molecule made in the lab is urea. It was actually discovered in 1727, isolated from urea. But how much urea we need today? For all fields and crops, everything, tons, right? Metric tons of urea you require. So is it possible that you isolate that much urea from the natural source? No. So here is the synthesis played a key role. In 1828, Frederick Wohler synthesized this urea in the laboratory for the first time, and this is the first molecule. And from here, the synthesis era began, and now we can able to produce metric tons of urea with the available chemicals. Then how much camphor we burn every day in temples and many places, homes, all those. It is again in ton scale. But this was also isolated from a tree called alpha pinene, which is abundantly in the oils of from trees, coniferous trees. But again, in 1903, Compass industrial synthesis of camphor. So this is from another easily available natural product, pinene. It has been synthesized, and that's how synthesis plays a key role. This is another important milestone in the synthesis. And just I am showing little complex structures also can be made, not only simple structures like urea and other things. This is modern era, you can think of total synthesis. How Woodward made a quinine in laboratory. So this was also a natural product which I have shown in a couple of slides back, and then now you can see this is being made in the laboratory. And these are several examples. Any molecule, if you plan a synthesis properly with available raw materials, one can make in the laboratory. And later it can be scaled up as per our requirement. And after this is the 20th century, now lot of reactions have been discovered, lot of technologies, lot of other things have been available. So from then we are also able to make various complex molecules, even having chiral centers, we can make the required compounds. So this is the era of organic synthesis, and Professor E.J. Corey in this picture is from Harvard University. He got Nobel Prize in 1990 for development of the theory and organic synthesis. So this is then why we are making molecules today. I have already mentioned that it is for large quantities and also for correcting the structures, and also it is inspired to develop a lot of methodologies. So these are the pioneers throughout the world making a lot of complex molecules, and in the present generation the down photography as Baron is doing wonders in making molecules. So there are a lot of molecules changed the world, particularly in health sector for identifying pharmaceuticals, and that synthesis really helped many compounds into the market. And so you see how complex structure this one. Today, scientists are attempting to make this molecule, even alphabets also not sufficient to number this molecule. So that's how that many rings and so much complex molecule, but still with available chemical tools and reactions, one can make this compound today. So coming to IICT, what we do here. Our director already mentioned that we work in diverse areas. Not only organic synthesis, we have various other departments, which I will show in at the end. But what we do is we synthesize natural products and their analogs towards drug discovery. We also make heterocycles. And besides that, as our institute mission, we also work for process development and various other high values in APIs, active pharmaceutical ingredient synthesis, to help the pharmaceutical industries, and also it will be helpful for having a lot of technologies with our country. These are some of the structures. I'm not going into the details. So now coming to with this knowledge, how IICT played a key role in developing new compounds towards COVID-19. So as we are already aware that drug discovery will take 8 to 12 years once we have the compound. So the only way is now with the available drugs, whether one of these drugs can be utilized for COVID-19. So with that, as soon as it was identified, the virus in December, then slowly scientists started thinking that which kind of drugs we can really think. And once we know that it is a RNA virus, then what are the earlier old RNA viruses, and what are the drugs have been used for this particular virus treatment. Then from that, WHO also announced and various uh, kind of molecules might be useful for this COVID-19. In that connection, around 69 compounds were identified for the possibility of treatment of this COVID-19. Out of that, 29 drugs are presently in the market, but for various other diseases. And then can we repurpose those drugs, which is because all these molecules safety data and other data already been studied. That means people are taking for a different disease treatment. That means the drug is safety, toxicity, all the studies have been done, so that it can be repurposed if it can cure this virus. So a lot of clinical trials are also started. Then how we played a key role from IICT in this particular molecule synthesis? Because we know these molecules, suppose if you take favipiravir, it has been discovered long back in Japan, and they are using for some other virus treatment, like seasonal flu. When it comes, they will take this favipiravir. So now, but it is not available in the Indian market, or even for that matter, umifenovir. None of these drugs are available in the Indian market, and we don't have compound also to test whether it will work for COVID-19 or not. So what we need initially some sample. Only we know the structure, and immediately sometime in February, we scientists from IICT and from other CSIR labs, we have selected around 15 molecules, so that if we can make these compounds available, we can have the process or technology for these compounds, and then we can transfer to some of the industries. And that's how we started work initially on favipiravir, and we were able to complete this molecule within 4 weeks during the lockdown period by staying at the institute itself. Our PhD students and some other scientists put their efforts and made this compound with the available materials within India. That is also key point nowadays. Even if you want to make this compound, you need some starting material. So to have the starting material also, we should think it should be cost effective and it should be available, and we need to make these thousands of kilograms. So keeping all these things in mind, we have initiated the work and completed within 4 weeks. And presently, this technology we have already, our director mentioned, we have transferred to various companies, including one of the major pharmaceutical company in India, Cipla. And these are the tablets you can see in the bottle. These were made from the compound made in IICT, and presently they are under clinical trials. Hopefully, in couple of next 3 to 4 weeks, the clinical trial results may come, and after that, drug controller of India may give permission, approve the launch this drug in market. So this is one of the drug, favipiravir. Similarly, remdesivir, another antiviral. Actually, it is not a drug in the market at all, but Gilead Life Sciences at USA were discovered this drug earlier for Ebola, and later it was not approved for Ebola, but it was under cold storage. But their company revived some of the old molecules when the situation comes, and it was under found that it is working better, especially for the patients under serious conditions. And they tried some of the initial trials under emergency basis. The US FDA gave approval to give to some of the patients, and it showed positive results. And later it was now approved for COVID-19 treatment. And this also gave license. The Gilead Life Sciences gave license to companies in India also to make this drug and market, because one company cannot supply all over the world. So they gave in India, in Hyderabad itself, we have companies Mylan and also Hetero Drugs. They also got license for this. But once, even if you have a license, you need to have the skills and the process ready for synthesis, because it is 100 milligrams injection. You need to have enough quantity of the compound in hand. That is where this organic synthesis plays a key role. And again, IICT is working since March. Now it is more than 3 months. We are working on this. We made this compound and also transferred this technology to some of these companies, and they are now making this compound. And once it is ready again, we are hoping that within a couple of months, this will be also available in the market. These are some of the other molecules. Uvifenol, this is also has been used in Russia earlier. It was also not available in Indian market. And then we need to have the compound and make this compound in the laboratory. And this is another compound we made at IICT. So these three compounds, KSR IICT had made the technologies and transferred to the industries. And we believe that at least a couple of these molecules will be in the market in coming months. And the other two molecules down, which EIDD 2801 galidesivir, are also potential compounds for the treatment of COVID-19. They are also under clinical trials at various places. And our sister labs from Trivandrum and Lucknow, CDRI Lucknow and NIAST Trivandrum, these labs have made these two compounds process, and presently they are under scale up. Once the scale up is feasible, they will be transferring to other pharmaceutical, some of the interested pharmaceutical companies. So this is about the role of chemistry, particularly organic chemistry, organic synthesis, and how it played a role in the drug discovery. And then this knowledge, how it has been utilized when the situation comes like coronavirus and the COVID-19 situation, and how we made this. Just I have shared some of these things. I hope the students, the present students, understand that very glimpse, very in a short way, that crispy manner I have explained that what is the drug discovery, how it plays, what is the earlier olden days, how it used to be, then how the organic synthesis evolved, and today man can make any kind of complex molecule with available reactions. And then that is how we also handle this COVID-19 possible drugs. And this is of course just a picture of our research group, and this is where at IICT we are working in this building natural products laboratory. And just to show some of these students, like many of them are bachelor students, and to just brief about IICT. Earlier in 1944, actually before our independence, this institute was established by Nizams of Hyderabad. You can just think of that those days, Nizam King thought that this kind of laboratory is required and it will help a lot of ways. That is how now Hyderabad also became a hub for pharmaceuticals. Initially, because of these institutes, and we used to have IDPL, all these things made Hyderabad as a pharmaceutical hub. And later, once independence came in 1949, initial foundation stone was laid up. And then 1954, the Prime Minister inaugurated the building, and 56, the CSIR takes over, and it was named as a regional research laboratories. Many people still knows that IICT has a RR in Hyderabad. So the original, it was named was old regional research laboratories, but in 1989, it was see some of these pictures, and there could be no demarcation in scientific progress. That was the one of the comment made during the inauguration of building. Now, since that time, IICT has grown up, and we had lot various other departments. And in the last 75 years, we built these many centers: catalysis, lipids, we have chemical biology, discovery laboratory, genetic chemistry, fine chemicals, agrochemicals, pheromones, and of course the natural products lab. So these are the departments now we have been, and these are some of the facilities established at our institute to do all our activities. And this is one of the unique facility in the country we have. It is called a National Moon Bank, where we store all our molecules, synthesized molecules, So it is called the National Mole Bank. We can store in thousands of molecules in this safely, and whenever the new therapeutic target identified, with all these molecules can be tested for the new target, and we may found, because earlier days before this was installed here in almost now more than 10 years. Before that, most of the compounds we made in the laboratory could not store for long time. If you store our normal refrigerators, it may long for some time, but later it will be decomposed or something. So even molecule, whatever we made with lot of difficulty, is also a valuable source for drug discovery program. So that is how this is helping. And this inside the mission, it will be looks like this. It is completely operated by robotic sample storage, where we made all the compound store and with a barcode. And whenever we require a compound for particular test, then we can retrieve from this by giving this barcode number to the computer, and automatically picks up the sample and gives back for various tests. And this is how these compounds are being tested through high throughput screening at IICT to find new drugs. So with this, I hope some of the students might have understand that how the drug discovery it helps, and synthesis natural products, very broad perspective I have mentioned, but in particularly toward this COVID-19, all our laboratories have played very key role in last 3 months. Despite of the lockdown, we took a special permissions and working in laboratories day and night to address the issue, and that's how we could able to make these compounds in the laboratory. And other than this, there are various several activities who are going around that making.
Thank you, sir, for such an illuminating and enlightening session. It was a great pleasure listening to you. Our students will be able to understand the drug better and will appreciate the hard work of all our scientists behind their discoveries. Our students can think of joining these CSIR institutions in future. In the attempt to make scientific discoveries, every problem is an opportunity, and the more difficult the problem, the greater will be the importance of its solution. To seek truth requires one to ask the right question. Over to my colleague, Dr. Gomati, for questionnaire session. Dr. Gomati.
Thank you, Mrs. Rashmi. It was a deep insight into the topic, the synthesis and the action of the drugs. The great pioneer scientist Louis Pasteur had said, science is the highest personification of the nation, because the nation will remain the first which carries furthest works of thought and intelligence. There are some queries from the participants. I would like to present them to you, sir.
Yes, sir.
Sir, uh, Flavipiravir, the drug designed by the prestigious IICT, what are the trial animals usually for the drug trials? Hello, sir?
Yeah, please.
Shall I read out the question again, sir?
Yeah.
Yeah. Favipiravir, the drug designed from the prestigious IICT, what are the trial animals usually for drug trials?
No, this was not actually designed by IICT. As I mentioned, it was already discovered earlier in Japan, and they have been using for normal flu, seasonal flu things. And then since it also found that it is against RNA virus, it will fight. With that knowledge, we picked up this molecule, and then it might work because the present coronavirus is also related to RNA virus. But whatever we are talking today's clinical trials will be given to some of the, because all the trials have been completed. That's how it was already marketed in Japan. So for your answer to your question, the clinical trials will have different phases. Initially, it will use initially test on the rats, and other later it will come to the other animals, and then at the end also phase 3, phase 4 clinical trials they will also give to the human beings. So that is why it will take 8 to 12 years of time. But presently these trials are that this drug is safe for Indian population also, that needs to be proven with some of the registered patient, then only it can be approved into the market.
Okay, thank you, sir. What are the natural products from which the present day COVID-19 drugs are evolved?
Yeah, there are some of the, as I mentioned, there are 69 drugs, 69 compounds listed by WHO and various other scientific organizations that might work against this coronavirus to treat the COVID-19. In that, approximately there are 20 or 25 natural products are there. I didn't have exactly names of these compounds, but there are at least 20 to 25 are natural products are expected that may work towards COVID-19.
Okay. Sir, remdesivir, how does it affect and target RNA?
No, it's a the mechanistic way means when we plan that, we know the protein structure of the virus and how through the molecular modeling and other things, how it fits into the that virus particular protein structure. So once it binds, that virus will not replicate. That's how it stops. That's called, kind of lock and key mechanism when we study in the graduation or post-graduation level.
Sir, what is the process of already discovered drug to test against COVID-19? What is the process? That is the trials, different ways, process stages.
Can you repeat again?
Sir, what are the stages for the drug trials?
Okay, drug trials.
Yes, sir.
Drug trial, particularly for this COVID-19, we have to register the patients and see the first safety. Whatever we have listed and trying to make the drugs presently are already all those levels crossed. All the trial safety, toxicity have been studied for those molecules. So the present study will be the clinical trials on some of the patients, how they are reacting, as well as some of the healthy patients, they are safe.
Sir, bats are reservoirs for this COVID-19, but bats are not affected by this virus. So can it be implored that why bats are not affected by this virus and why human beings are affected?
I think the virus, maybe the host, for them, the best host is probably human being. That is why I think we are being affected.
Okay. Sir, one last question, which many of the participants wanted to know about the homemade hand sanitizer, sir. Homemade hand sanitizer.
Homemade hand sanitizer.
Yes, sir.
Hello.
Sir, homemade hand sanitizers. They wanted to know about the homemade hand sanitizer.
See, sanitizer is basically mainly at least 50% of either ethanol or isopropyl alcohol is required, so that alcohol will have that property to destroy this viral virus. Is a kind of small protein, so that it breaks the amide bonds in that when we wash our hands with alcohol. So any sanitizer you make, it should contain some alcohol to break the protein.
Yes, sir. Thank you, sir. Without your involvement, you cannot succeed. With your involvement, you cannot fail. So our great teacher today has patiently answered to the queries of the participants. Thank you, sir, once again. And over to Mrs. Rashmi.
Thank you, sir, for clear explanation. Thank you, Dr. Gomati, for efficient handling. These approaches resonate with the IICT's vision to serve society by creating an outstanding knowledge base in chemistry and chemical technology. Now, for a few words of gratitude to the people who made this webinar a success. It has been our pleasure to host all the elite participants of the webinar of Little Flower Degree College. The participants were very enthusiastic, and I'm thankful to all the participants for joining us to attend this webinar on role of science and technology in mitigating COVID-19. Today, we have been fortunate to have eminent resource person, Dr. S. Chandrasekhar sir, Dr. Raji Reddy sir for our webinar. The lecture delivered by our speakers were really informative, inspiring, and motivating. I'm sure that the participants must have benefited by today's lecture from our eminent speaker. Link to download e-certificate will be provided in the chat box, followed by feedback form. I request all the participants to download the e-certificate before leaving. Now I request Dr. P. Dharna from Department of Zoology to render vote of thanks. Dr. P. Dharna.
Dear participants of the webinar, on behalf of Little Flower Degree College, I take this opportunity to express our sincere thanks and gratitude to the renowned scientist of our country, Dr. S. Chandrasekhar, Director CSIR IICT, and Dr. C.H. Rajareddy, Senior Principal Scientist, CSIR IICT, for accepting our invitation to speak on the role of science and technology in mitigating COVID-19 in this webinar. The lectures delivered indeed by our speakers were very informative, inspiring, and motivating. I thank them for sharing their immense scientific knowledge and technological know-how in mitigating the effect of COVID-19 and for their fruitful interactions with the participants. Friends, when disease outpaces containment, countries rely on mitigation strategies. We believe that we shall overcome this pandemic soon, as India is blessed to have esteemed scientists such as Dr. S. Chandrasekhar and Dr. C.H. Rajareddy, whose contributions may lead to unravel the COVID mystery. I thank all the participants for their active participation and cooperation. I'm sure that this webinar was beneficial in updating their knowledge. I sincerely thank our college management, principal and vice principal for their constant encouragement and continuous support for conducting these events. I also thank the staff, faculty, and students of LFDC for their kind help and cooperation in organizing the webinar. Finally, on behalf of Department of Science, I once again thank everyone whose contribution had made this national webinar a grand success. Thank you one and all. Have a nice day. Over to Rashmi.
Thank you, ma'am. Some days are better, some days are worse. Look for the blessings instead of words. Be positive, stay strong, and get enough rest. You can't do it all, but you can do your best. Thank you, one and all. Stay safe and be healthy. Mrs. Rashmi Srinidhi, Head Department of Chemistry, signing off. Good afternoon.